components, histological aspects of tissues don't look generally that blobbish and non-uniform. So this is diagnostic. This and this plaque and these plaques and then these tangles, neurofibrillary tangles. These are diagnostic of Alzheimer's and other neurodegenerative disorders. Unfortunately, this can't be diagnosed, at least currently. I think there's some promise for it, but up until very recently, this is post-mortem. But it's very clear, very compelling evidence that these things are what are interfering with function and in a progressive way. Now, first of all, we're going to talk about Alzheimer's and some of the clinical symptoms, not so much that you'll need to know details of the clinical symptoms, but you have to understand some aspects of it in order to understand the significance of the research. Okay. And what I wanted to just apologize that there very well could be somebody in this room who has a relative grandparent that has Alzheimer's or some other dementia. And I'll just tell them so I don't need to be insensitive at all by any means, but we do have to talk about the clinical aspects. And I'll just let you know my mother passed away recently with dementia, not Alzheimer's, but it's sad to see somebody going through that. She was 91, so it wasn't too surprising that she lost some of her cognitive functions. But on the other hand, there are people that live to be in their late 90s and early 100s, and they retain essentially all cognitive. They probably have more cognitive functions than I have. I feel like I'm starting to lose some, and I'm only 63. Young, 63, right? So, but yeah, so this is, now I did assign this review article. We just wanted to get some aspects of it. I don't have the study guide finished quite yet, but I will have it up this afternoon. You're not expected to know everything in that review article, so go by what I have in the study guide and go by what I'm emphasizing in the slides. Okay, but this is the paper for tomorrow. But wait a minute, this isn't even one of the genes that's responsible for the heritable form of Alzheimer's. So there's two very distinct, as we'll see, inheritance patterns for Alzheimer's. There is an early onset, Mendelian, monogenetic, autosomal dominant Alzheimer's. Okay, and that's early onset, like people in their 20s and 30s, I believe. And it's autosomal dominant. I can't remember if there's complete penetrance or not. But we'll see that there's actually 3 different genes, very directly related in their function, that are responsible for those monogenic forms. But those are rare. The most truly monogenic disorders in humans. This is actually getting to do with what's much more common, and that's the sporadic forms. Now we know a lot of the susceptibility loci. There have been a number of GWAS studies. I'll show you the main conclusions from some of those. But even before we had GWAS, strictly by linkage analysis, ApoE was shown to be a real strong risk determinant for Alzheimer's, for the sporadic forms. Now I don't know, I can't remember now if I'm not sure there would be any reason to look at any association with that ApoE with the familial forms of Alzheimer's. I'm not sure, but anyway, this is focused on that. And it uses CRISPR and iPSC cells in a really cool way. Now I had already assigned and uploaded 3 review articles. This is another one, because when I was looking at everything yesterday, I realized I hadn't given you enough background information to ApoE. Okay, and that's what you can find. I just uploaded this today. Now again, you don't have to. If I've got any figures, and I've got, I think I've just got extra figures from here in the slides. Just look at, if you want to know more information than what's contained on the slides and what we talk about in class, just pick those out from the review article. So you do have to understand a little bit about what ApoE does in order to understand its relationship to Alzheimer's. Okay, so first of all, I just want to give you some let you know some facts, and you're not gonna be tested on these, but I just wanna get across the significance. This is a truly significant disorder in humans, okay? And I took all of these, the next few slides from the Alzheimer's Association site, and they update this page, these facts about Alzheimer's every year, and I compared the facts, the details of some of these, what I've got on these slides from 2019 to last year, and even in 1 year, there's an increase in the numbers of people that are being affected by Alzheimer's. So of course it's a, clinically devastating disorder to the person, but as you can probably imagine, it's pretty devastating to family members and caregivers for people like that. And then we've had, I hate to put, economic values on a devastating, both physically and emotionally disease, but it is tremendously expensive as on the whole. Now, there are some other susceptibility things, so age, of course. And, just look at this pie chart. It's mainly a disease of older people, okay? But look, I'm in that age, 65. Okay, there's quite a few people. I'm only 2 years from now, I'll be 65. There's also, I'm not sure, I don't know enough details to know if there's an explanation for this sex difference. So females are much more susceptible or more likely to be at risk for Alzheimer's. Now, some of that could be just that women tend to live longer than men. At average age, it is more in women. So part of that, but there could also be some basic sex differences. This is what I find really scary. The projections based on the current, kind of rate of diagnosis. This is, the rates projected up to 2050 aren't changing so much in this age, changing a little bit and slightly older. This big increase, anybody have any idea? I mean, these numbers are a little bit misleading because they should be normalized. This is total millions of people with Alzheimer's, and 14 million people expected by 2050. That's not that far away. Where this is a little bit misleading is that this is not normalized to the total projected size of the population in this age range. And anybody know a particular, baby boomers? Yeah, baby boomers are getting older, and I'll just tell you, I was born in 1955. I'm right smack dab in the middle of baby boomers. So, I'm in this range. I'm not sure I'll live until the 2050s, so that would be pretty cool. I guess I'd be 94. So I'm not sure I told you this before, but there's a really helpful internet site database called Online Aliens and Man, OMAN, where you can go, and there's a link to this, and there's a specific number associated with every human genetic disorder that's been characterized at all, and it's not even genetic disorders where the gene is known. It's any genetic disorder in humans. If there is a gene known, then they have a number for the gene, and then have a number for the disease, and they're linked. If there's multiple genes, then they're all hyperlinked. This OMAN, this database tends to be up to date for some things, but don't count on it being up to date. Look at the references to see. But there'll be all kinds of hyperlinks, and it'll give a good description. Probably more than what you want to have, but if you ever wanted a starting point on looking up some genetic disorders. So there are several other forms of dementia, I think you probably know of, but this is the most common form by far, up to 80%. And there's a loss of memory and cognitive functions, and there's all kinds of behavioral issues associated with it. People can live, so they tend to be diagnosed, for the sporadic forms, tend to be diagnosed in their 60s or so, and some people unfortunately don't live very long after the diagnosis. Some people can live for as long as 20 years, but it just kind of all depends on their situation, their general health status, whether they've got some of these susceptibility loci. But where the research is directed, now currently there's no treatment. There's some ways of slowing down and lessening the severity of the symptoms, but there's no treatment. So there's still a lot of active research. And basically the pathology in terms of loss of cognitive function is because of death of neurons relating to the abnormal formation of the plaques and the tangles that were on that first slide that we'll talk about in more detail. Now, these are age, gender, family history are the most significant risk factors. I told you about just less than 10% of the patients with Alzheimer's have one of these three possible mutations. Okay, so this is that genetic heterogeneity where the same disease can be caused by mutations in different genes. Okay, but we're going to mainly be talking about the non-Mendelian ones. The advantage, though, of having both Mendelian forms and non-Mendelian is that once the genes were identified that are responsible for the Mendelian forms, even though it's only a small number of patients, it's still been incredibly useful in understanding the pathogenesis of this disease because the genes that are defective here are all related to the processing of one protein. Well, two of them are, and then the other one is the actual protein. And so that gave really good clues about what to look for in terms of things that are disrupted with the late onset. So, yeah, don't be thinking that just because Mendelian forms of a particular disorder are really small, low in frequency, if there are sporadic forms of the disease, the Mendelian forms can still be very useful biologically. Okay. Now, non-Mendelian, but there's a huge, large genetic component, meaning environment can affect. Okay. The paper that I assigned uses SAD, spontaneous Alzheimer's. Other papers you might see if you looked at them refer to this as being low for late onset. But either way, these are the non-Mendelian, polygenic, affected by environment, multiple genes. Okay. So this is all animation, but it's from the NIH, from the National Institute of Aging, and so I think it still will give you some important information, and you're encouraged to watch it on your own, because it tells you, it describes some of the cell types that are involved, and it describes how those plaques and tangles are forming, and how they might be affecting neuronal function.
In healthy people, all sensations, movements, thoughts, memories, and feelings are the result of signals that pass through billions of nerve cells or neurons in the brain. Neurons constantly communicate with each other through electrical charges that travel down axons, causing the release of chemicals across tiny gaps to neighboring neurons. Other cells in the brain, such as astrocytes and microglia, clear away debris and help keep neurons healthy. In a person with Alzheimer's disease, the most basic form of dementia, toxic changes in the brain destroy this healthy balance. These changes may occur years, even decades, before the first signs of dementia. Researchers believe that this process involves two proteins, called beta amyloid and tau, which somehow become toxic to the brain. It appears that abnormal tau accumulates, eventually forming tangles inside neurons. And beta amyloid clumps into plaques, which slowly build up between neurons. As the level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. But tau and beta amyloid may not be the only factors involved in Alzheimer's. Other changes that affect the brain may also play a role over time. The vascular system may fail to deliver sufficient blood and nutrients to the brain. The brain may lack the glucose needed to power its activity. Chronic inflammation sets in as microglial cells fail to clear away debris, and astrocytes react to distressed microglia. Eventually, neurons lose their ability to communicate. As neurons die, the brain shrinks, beginning in the hippocampus, a part of the brain important to learning and memory. People may begin to experience memory loss, impaired decision-making, and language problems. As more neurons die throughout the brain, a person with Alzheimer's gradually loses the ability to think, remember, make decisions, and function independently. Achieving a deeper understanding of the molecular and cellular mechanisms and how they may interact is vital to the development of effective therapies. Much progress has been made in identifying various underlying factors. Advances in brain imaging allow us to see the course of plaques and tangles in the living brain. Blood and fluid biomarkers are providing insights about when the disease starts and how it progresses. More is also known about the genetic underpinnings of the disease and how they can affect particular biological pathways. These advances enable the development and testing of promising new therapies, including drugs that reduce or clear the increase of tau and amyloid proteins in the brain, therapies targeting the vascular system, glucose metabolism, and inflammation, and lifestyle interventions like exercise or diet, and behavioral approaches like social engagement that may enhance brain health. Research is moving quickly, ever closer to the day when we can delay or even prevent the devastation of dementia.
This video referred to 3 cell types that are important to the assigned research paper. They were able to study, they were able to make iPSC cells, differentiate them into neurons, differentiate them into, you don't want that one. Differentiate them into neurons, astrocytes, and microglia. And you don't need to be a neurobiologist in detail, but I do have some explanations about those, and you might want to read a little bit more about them if you're interested. I'm just telling you the general aspects of them. But it is important. I mean, with any tissue, there are multiple cell types in a tissue, and it's really can be misleading in terms of trying to get a good assessment of disease mechanisms if you're only looking at one cell type. Where did it go? Oh, okay. So please do watch the animation again, because it will be helpful to you, I think. So the video referred to these plaques, and amyloid plaques. They're made from, we'll see how this precursor protein, APP, amyloid precursor protein. It wasn't even, as I recall, I don't think it was even known. I think the first evidence or first suggestion of this protein came from identifying the genetic basis of one of the familial forms of Alzheimer's. Okay. But as we'll see, the 3 Mendelian forms of affect the processing of this protein. One is actually the protein, and then the other 2 affect the cleavage. Okay. Now this is where we come in with the disease susceptibility, ApoE. So this is apolipoprotein E. So this is like LDL and HDL. It's a carrier of cholesterol. Okay, so the gene encodes the protein component of it, but then cholesterol is synthesized only in certain cells, but it's got in the bloodstream, it's carried through the blood. That's why your doctor is looking at your LDL levels and your high and your HDL levels. Okay, the low is the bad and high is the good. But in the brain, ApoE doesn't. Okay, now ApoE is also functioning to a certain extent, I think, in other in the blood system too, and I'm just not sure what the significance of that. But this is what actually binds one of the cleavage products of this precursor protein called A-beta. So the beta amyloid plaques comes from this A-beta. Beta is one of the small cleavage products of this precursor protein. And they know what the composition of those plaques is, that is made almost exclusively of A-beta peptide, but they don't know still what's causing that aggregation. There's becoming some clues. Now, the other pathology here are the tau protein. Now, I don't think the video mentioned this, but this isn't just regular old tau. It actually gets hyperphosphorylated. So something, and they've got some clues, something is causing that. Normally, that tau protein is essential for function, normal neuron function. But something is causing it to be, well, as I say here, hyperphosphorylated, forming those tangles. Okay, and both the plaques and the tangles are contributing to the overall pathology. Yes?
For the ApoE and the A-beta protein, is one of them sort of like, does one of them come first, the accumulation of the ApoE, like that binds to that protein, the cleaved one, or does the cleaved protein?
We'll see this ApoE actually, so the APP precursor protein gets processed by a couple different enzymes, okay? And normally the A-beta form is not produced in high levels, but it is for a number of different reasons. But that A-beta is then picked up by ApoE and delivered normally to other cells, okay? And so the A-beta is going to be soluble in the extracellular space. ApoE is going to be in the extracellular space. Then that, we'll see, this is getting ahead, but then that complex of ApoE and A-beta are delivered to neurons, and delivered that way through endocytosis. So that's the normal process, yeah, but something is going haywire with this. Did that answer your question?
Yeah, so when you say like something is going haywire, that they're not getting delivered, or that there's just a lot more?
There's a lot more, or they're accumulating in the wrong place. You'll see there's some, I think some things will become clear with what you're thinking about. So these plaques, I can't remember, I think it was, this is where you can look in the Tansey et al., the assigned review article. It'll tell you more about the history of it. But as soon as they identified these plaques, and remember at that time it was all based on post-mortem analysis, and then they figured out that it's because of abnormal accumulation of beta amyloid, then they came up with what is still called the amyloid hypothesis, that Alzheimer's is caused by that accumulation of handling of this amyloid precursor protein. Now, some people say this doesn't explain everything about it. Other people think it's still a good kind of overall. It does, it's not meant to explain everything about it. So if you started reading a lot of the literature on amyloid and Alzheimer's, you might see people kind of dismissing this hypothesis. But I think overall it's still considered to be a good hypothesis. Again, not explaining everything, but explaining a lot. So this is that same slide from the first, same image from the first slide, but now I'm showing you normal. And it's more obvious. There is a lot of things distinctly wrong with the brain section of the left. So they'll say, yeah, these are the plaques and neurofibrillary tangles. And what they do then is cause cell death to the neurons. And that's a problem. You want to keep all your neurons. You don't want them going. So this is showing some of the same things that were alluded to in that animation. I just took them from a different site. But you can follow both in terms of the clinical symptoms and with later stages, what's happening in the brain morphology. But there is a pretty characteristic progression of events, both happening at the level of the brain and the cells and with behavior. So some of these changes you know, they could be happening in me right now. I'm not aware of them, but maybe, you know, every time I forget something or a simple saying a word, I think, oh my God, these are early symptoms. I will tell you, I am heterozygous for the susceptible allele of ApoE. I don't know, my mom had her DNA tested, but she didn't have it done through 23andMe that would have told us the alleles that she had. So, but, you know, I've gotten this far. I think I'm doing okay. And besides, I'm still going to try to stay healthy and watch my have a good diet and exercise and watch my cholesterol level and keep intellectually stimulated. Okay. So, but there's some early things that can happen, and I don't think they can really diagnose people because you've got to make a distinction between just normal age-related, you know, kind of loss of memory and forgetfulness and, you know, things like that. But I think in retrospect, they can see some of these things happening. Now, once it progresses to the mild to moderate, then you start seeing much more of the pathology. You start seeing a lot more behavioral issues. Okay, in this speaking and understanding speech, my mother completely lost she couldn't articulate anything other than like single words in the last like 6 months of her life. The sense of where your body is in relation to the surrounding, there's a kind of clumsiness and just, you know, they lose the ability to kind of know how far they are from objects, and you know, so, and this is still a pretty mild to moderate, but notice the blue area. So this is where the region of the hippocampus, and I don't know if they know I don't know why it's specific for those particular cells in the hippocampus. Does anybody know? Does anybody take any neurobiology or know anything more about Alzheimer's? You could probably look it up if you want to find it out. I think it'd be really interesting to know. But the point here is that it does have a very specific place of origin where the earliest signs are seen, and then it spreads from there. Okay. And have you all heard of prion diseases, where the protein is actually spreading? Yeah, there's a lot of people that, I think there's some evidence that those plaques are considered like prions and that they move from cell to cell. So they're extracellular, but they're the basis of spreading throughout the brain. So now this is advanced. And then once the brain starts looking like this and people start getting, this is where they lose the ability to communicate, they don't recognize family, they stop taking care of themselves. And this is where the caregivers come in. And it is a big burden, I know that with my friends have been, you know, the caregiver for elderly parents with Alzheimer's. It's not a task anybody wants to have, but you know, nobody's going to say no to do it. You gotta do it. But yeah, so this is where the cells start dying. Now look, this is, you know, kind of taken on the head. One half of the image is a postmortem brain slice. The other half is a healthy. All of these spaces are basically because of loss of cells, because of cell death. This isn't even showing anything with the plaques and the tangles. This is just how small the brain gets when it starts getting so severely affected. You don't want to have a brain like that, trust me. Yeah. And this is what the video referred to with the metabolic changes. This is MRI, and there's probably better imaging techniques now than this one, but they can tell that there is. And now I don't know, again, this is where my knowledge of the details of Alzheimer's isn't that detailed. I don't know if they're thinking that the altered metabolism is a consequence of the other pathology, or if altered metabolism is contributing to the pathology. Either way, it's pretty diagnostic. And I think they are developing or trying to develop some diagnoses based on MRI and other kinds of imaging. Or this is a PET scan. I don't know the difference. Sorry. Okay. More about the protein. Okay. And the genes involved. Now, so first of all, is the familial forms. As I mentioned a few minutes ago, they're all autosomal dominant. Oh yeah, highly penetrant. They were identified through traditional linkage, positional cloning, back in the old days when it took years to positionally clone a gene. But they discovered that's where this amyloid precursor protein was discovered. Okay. And they know that this is on chromosome 21. This is the protein, the precursor protein, right, that gets processed. And there are specific cleavage sites for specific enzymes, alpha secretase and gamma secretase. Well, so not too surprisingly, the two other familial forms of Alzheimer's have mutations that affect the actual enzymes that do that cleavage. Okay. So really, right away, people knew APP and its processing was key to understanding Alzheimer's and pathogenesis. Okay, so I'll be careful in the study guide just how much of a detail about the APP protein, but you do need to know some aspects of it. So this is the precursor protein. It's a huge protein, right? This is the inside of the cell. This is the outside of the cell. Transmembrane domain. This is the actual what's referred to as being the beta domain. This is what's in red. This is showing the positions of the 3 cleavage sites for those 3 specific enzymes. Alpha secretase releases the extracellular domain. Beta secretase then makes a cut in that released extracellular thing. Gamma secretase leaves here, and then this green part ends up going into the nucleus and affecting transcription of genes in the pathological aspects of this processing. So there's a normal type of processing, and then there's a processing that takes place that causes the disease, Alzheimer's. Okay. And the same things can be seen in terms of the abnormal processing and handling of particularly A beta that we'll see, one of the cleavage products. That's common to both the familial and sporadic forms. Okay, so again, it's very clear this is the key player in the pathogenesis. Okay, so this is the non-pathogenic pathway. So first there's cleavage here in the A beta domain. This is the precursor again, like we saw. That releases this protein soluble. Now this is an essential protein. If you do a knockout, make a homozygous null mutation in the mouse, I think they're embryonic lethals. I don't think they're even born. So APP, and particularly the soluble form of it, is absolutely essential for proper brain development and function. Okay. Then there's additional processing going on. Here's where you can see it here. There's alpha secretase, gamma secretase cuts there, making this. I forgot what AICD. Amyloid. Anyway, it's an acronym. You don't have to know about AICD, but normally that's going to be degraded. We'll see there's a very different outcome in the pathological state. Okay, so this is normal, everything going on here. Basically, the most important processed polypeptide is a soluble APP alpha. Now this is the pathogenic. Okay, you still get cleavage there, and you still get cleavage here, but it's slightly different. Okay, there's actually 2 different. That soluble is a little bit different, and this other cleavage product, it just differs by a few amino acids. Okay. So, but now because of the slight differences in the cleavage, this protein is actually being active and combined with this, and it goes in and affects gene expression. This is in the pathogenic. Then it's this guy, A beta, that it does differ. I mean, these are the same amino acids here, but it's different enough, and when it's produced at high levels, that's what's causing the problems. And you might see it in the assigned paper referring to A beta 42 and A beta 40. These just differ. There's 2 different sites here that just differ by 2 amino acids. So this is a 42 amino acid polypeptide. This is a 40 amino acid polypeptide. So these little bit, and I think both of them can contribute to the plaques. So these pretty small proteins are making those big plaques. But what they're doing is aggregating it normally. Okay. And a lot of people are saying that it's I don't think they understand why this ratio is important, but that's something that still needs to be investigated. Now, we're not gonna be looking at all, I don't think in this paper. They're not, I know they looked at gene transcription. I'm not sure if they were relating it to AICD or to other aspects of the pathology, but I'll have that in the slide if necessary. Okay, now again, back to the familial form so that we can understand more of what's going on in terms of processing of the APP. Okay, so just so you know, there's lots of different mutations in APP, including a duplication. And the three things happen with APP. Now, a lot of these mutations, a lot of them are missense mutations that are specifically at those sites of cleavage by the beta and alpha and gamma secretases. Okay, so if you change that cleavage site, you're changing the processing. The ratio is altered with APP proteins. Remember, these encode the secretases. They all are associated with different ratios of A beta 42 to A beta 40. This has increased A beta production and aggregation. Now, I want to point out something here. We won't talk about it further. You think there's any significance? Can you think of anything biologically connected to APP being located on chromosome 21? Can you think of any other well-known? Yeah, it's Down syndrome. They often have cognitive, and some are age-related. They have 3 copies of APP. Gene dosage with APP is important. Okay, now we're getting into the late onset or sporadic SAD forms. And there's a lot of genes that have been picked up, and you should be automatically reviewing in your mind how genome-wide association studies are done. I just covered those. ApoE had already been identified by linkage analysis before G was to be a risk factor, but it was very encouraging that it was reproduced in these G1s, along with other genes that probably have smaller effects. So as far as I know, ApoE has the largest effect on the risk. In addition to genetic, in addition to sex, age. Yeah. Okay, so what is it about ApoE? Well, it's not ApoE itself. It's the fact that the natural variation that exists in the human population, there's actually 3 SNPs. Well, there's probably more in the ApoE gene, but 3 of them that change amino acids. Okay, and they're not subtle changes. I mean, they are kind of subtle changes, but they do have a very pronounced difference in function. Okay. So the 3 alleles are E2, E3, and E4. And actually, what's interesting is that E2 seems to be protecting against Alzheimer's. E4 is the risk, increased risk. Then E3 is intermediate, so there is a dosage effect. If you're homozygous for E4, that's a real high risk. Heterozygous isn't too bad, but it's not a huge, I'm heterozygous for E4 and E3, and I'm usually concerned about it. If I was homozygous, I probably would be concerned. But I don't remember if heterozygous for E2 and E3, if there's a dosage there. I do know homozygous for E2 is protected with much lower frequency. Okay. They do these proteins that are encoded, right? They differ just at these 2 positions, amino acid 112 and 158. Now remember, this is not APP, this is ApoE. So far we haven't yet seen the connection between ApoE and APP processing, but we will, before we do that, let's take a